So the protein structure is given to us as really this geometric. It contains geometric information, very high resolution. We know the coordinates. If you look at these N, C atoms, the nitrogen, alpha carbon, the other carbon, and the oxygen, the beta carbon, so carbon beta, carbon gamma, delta, so different amino acids. For example, proline has this other additional carbon atoms in its residue part. These large atoms are also given there. So as you can see, they are given at each atom is given by a coordinate, and this coordinate system is really, as I said, is something arbitrary. It's you may translate, rotate your protein. These will preserve the protein structure shape. Okay, so this translation and rotation are rigid body transformations, which will not make your protein structure shape different. If you scale it, for example, if you want to scale it down, it will no longer be a natural biological shape, because the bond lengths, the atom positions, they will no longer represent the true distances in nature. But translation and rotation, it's really does not do anything as to the protein structure. It's just locating it and rotating it in a different place. So this position here, it's really not relative to some cell. It's not, for example, relative to the nucleus or the chromosomes, or it's not relative to the cell membrane. So these coordinates are really arbitrary coordinates with some local reference coordinate frame. So there are different motifs. We have seen for example, the zinc finger domain is like a small structural shape, which may be important for a specific function. And there are also, we see, if you look at these protein structure, there are certain small common structural shapes, formation of these 4 helix bundle, which is apparent in many protein structures, which may serve for a specific function. These are called structural motifs. A motif is a certain shape, a certain small substructure, which is observed more frequently than random occurrence. Such things are called motifs. And if you study structural bioinformatics, you will see that these motifs are studied too. Secondary structure prediction, I'm not going to go into too much detail about secondary structure prediction. As I told you, it's a one-dimensional structure prediction method, and it's the first step, most of the time, the first step in 3-dimensional structure prediction. So the problem is, given an amino acid sequence like this, our goal is to classify each amino acid into one of those three categories. E is for beta strands, H is for helices, and C here is for coils or turns. So we classify each of them to one of these 3 letters, C, E, or H. And this is that prediction problem. And as you can see, there may be some prediction errors in this example, because 2 amino acids, they will not be able to form a helix by themselves. So because the formation of the bonds is between every I and I plus 4th amino acids. So this is probably a wrong prediction. As you can see, probably the C's in the middle here, they may need to be turned into helices. Also, this is a really short chain. You probably do not see the other part, because again, these beta strand itself is not going to form a beta strand on its own if there is no other beta strand somewhere else in the sequence, because they can only form beta sheets if at least 2 strands are needed to do that. Secondary structure is the first step, as I said, to 3-dimensional structure prediction, because after we find these, then the next step is how they are oriented. These turns that are in between, you can use them as a degree of freedom. I mean, you can have more rotation in those turn parts to orient these secondary structure with respect to each other. So you may have different three-dimensional structure prediction methods by after you find the secondary structure first. It's an easier problem than 3D structure prediction. It's mostly a solved problem, although there we have like more than 90% accuracy in helices, helix prediction, and close to 90% prediction maybe for beta strand these days. So secondary structure prediction is mostly a solved problem. Yeah, it can also be used to improve sequence alignment accuracy. If you perform sequence alignment and you align two parts of two very different sequences, you may use these secondary structures as your guides to make small corrections in your sequence alignment. And there are different prediction methods. There are SciPred, JPred, which use neural networks. They are nearest neighbor based methods. So there are many heuristics, many different machine learning algorithms, some statistical methods. There's GORE 1 to 4, or some machine learning methods are used to predict the secondary structure. I'm not going to go into details. The statistical ones, they usually slide a window of certain size, 13 to 17 residues, to find those propensity, this amino acid preference values. That's the Cho-Fosman method, not the GORE. So you compute the parameters for the amino acids to be in alpha helix, beta sheet, or turn. They just sum them up using a slide method, and they have some built-in rules. For example, if you have a region where 4 of the 6 amino acids in the window have more than PA more than 100, then do this, then extend it this way. So these are really hard written rules, but they're basically doing this by using those preference values. So you're not responsible for secondary structure prediction methods in the exam. Therefore, I'm going to skip this one. And just look at this table from a paper by CyPred, I remember. So this is from a 2004 paper, a simple and fast secondary structure method using hidden neural networks. I think that was CyPred. And it shows you that there are different scores. The helix prediction score, this one is around 80% accuracy, but as you can see, the beta strand prediction is relatively lower. The prediction accuracy is lower for beta strands compared to helices or coils. But this is mostly due to the fact that beta strands are due to interaction between amino acids that are far in the sequence. They are not really local. They also have some global structural properties that makes them difficult to predict. These days, I think this bar is just raised, I think, to 90% these days. Since 2004, I think for example, if you search for protein secondary structure prediction accuracy, let's take a look at I would be surprised if deep learning was not used to predict secondary structure. So they have a Nature article by using this Nature methods, scientific reports. So they report deep convolutional neural fields. Basically, this is deep learning. And they also, it's not that high accurate actually. They have 80%, so they were able to increase the bar to 84%. So it's not as high as that I thought. Okay, so the accuracy is still 85%, around 85%. And this was a recent article, right, in 2016. Not so recent, but in five years ago. This article was published in Scientific Reports. So it's still, people are still doing research on secondary structure prediction. So there is still room for improvement. All right. Now, finding structure is really important. The first, if you remember, the first months of the COVID, the pandemic, uh, the COVID, the virus proteins were identified. So it's the we have identified its genome. You may, if you call it like that, but the proteins, the set of proteins were identified, and there was a nice article about this. I made a post in call. If you remember last year when I was giving this course, I made a post about this. I may find that. Let me just search for here. COVID 465. No. Oh, maybe this one. Yeah, right here. No, not this one. Coronavirus. Or maybe I can just go to, no, I couldn't find it. But yeah, I can find that website and submit it again and not to here. But let me just find it quickly. So last year it was this. Yeah. So let me look at the announcements I made last year. Yeah, the proteins of the coronavirus. So I found it. So look at this. So I will make the same announcements in our OT class announcement page, but just I wanted to show you here now. In New York Times, so this was a New York Times article. Bad news wrapped in protein inside the coronavirus genome. If you look at that, so the first thing they did was a string of RNA, then the sequence of RNA letter reads, so then the chains of proteins. So they looked at which proteins the coronavirus genome makes. So this is one of them is, for example, cellular saboteur, okay, NSP1. So this is its sequence. This is a structure. It's as you can see, all of those structural. These are protein structural shapes. The first thing they did was to identify for example, this is mystery protein. Scientists aren't sure what NSP2 does. Maybe they do now. So this has been 1 year over this article. But the other proteins it attaches to may offer some clues. So these protein shapes, three-dimensional shapes, as you can see, the molecular surfaces are shown here. They interact with other proteins. So interactions of proteins with the other proteins are also really important. We are going to talk about these biological networks at the final weeks of this semester. But this is a really nice article for untagging and cutting, which shows just outlines all the proteins of this virus and what it does and how it hijacks the host cell. So NSP3, it's a large protein that has two important jobs. One is cutting loose other viral proteins so they can do their own tasks. It also alters many of the infected cells' proteins. So it's, as you can see, there's different shapes. Bubble maker. So they gave them nice names like this. Combining with other proteins, NSP4 helps build fluid-filled bubbles within infected cells. Inside these bubbles, parts for new copies of the virus are constructed. Protein scissors, so cuts other free NSP proteins. Another one, bubble factory, copy assistance. In order to copy the virus to copy itself, these are, as you can see, this one has a really different shape. This one, too. They two together at the heart of the cell. So this is also like a finger here, as you can see, probably very important to its function. Genetic camouflage, copy machine, which is a larger protein, as you can see. Unwinding RNA, viral proofreader, cleaning up, more camouflage, spike protein. You may have heard this protein a lot. Most of the vaccines here, some of them, they use the spike protein a lot when developing the vaccine. So it's one of the 4 structural proteins that form the outer layer of the coronavirus and protect the RNA inside. So this is probably how our cells recognize if and produce the anticore for this virus. So spike proteins are therefore important. And they have a very different shape, as you can see. There are these different spike proteins, and they're very large. Escape artist, envelope protein, membrane protein, signal blocker, virus liberator, another mystery one, and then finally combine all them together, you have your coronavirus. Okay, so I'm going to share this article with you on the announcement. So it shows you that finding the structure is really important. For example, if you know the structure of that spike protein, you can then develop certain things that will resemble that. So structure is really important to develop medicine. First understand its function, how it works, then develop countermeasures to block those proteins or to have our body naturally construct a defense mechanism against those viral proteins. So structure function, we know that they're really important. They're structure-based drug design. So this is also another field. In our own bodies, sometimes our own proteins are making us sick. So most of the drugs that are available in the market are designed in such a way that they block our own proteins. The body, if the certain cells malfunction, for example, they start to produce a certain type of protein in excessive amounts, and or the functioning of that protein may make you sick. So if you design a drugs are basically small chemical compounds like this, which interact with a target protein and blocks its function. Okay, they are most of the drugs. This is really when I first learned about this was really counterintuitive. I mean, when you take a drug, which makes you well, it's like a positive synergistic effect, right? I mean, it makes you well. So you think that is something that boosts something in the body. But it's actually the reverse, actually. So all of the drugs are like blockers. They interact with protein and they block its function. And most of the time they block our own functions, our own proteins functions. So one research area is how to design those drugs, meaning that, for example, if you know the protein structure, which chemical compound, well, how do you design a chemical compound that will interact with that protein's active site and block its function? This is really challenging, and there are many factors that are involved in this. For example, okay, you may have found a very nice chemical compound which is going to interact with that protein and block it, but what if it blocks some other proteins too, which will produce side effects, basically? How do you minimize the side effect of a chemical compound? Design a chemical compound in such a way that it will be taken inside the cell by the cell. You cannot make your chemicals too large. They have to pass through those ion channels. And at the end, they should have a specific target protein, and they should not interact with too many other proteins. Otherwise, you may have other unwanted effects. So therefore is really challenging finding such chemical compounds that interact with protein surfaces. This is the techniques that are used in this is called docking, protein docking. You basically have it's again structural problem, but you have a chemical interacting with a certain part of a protein, like a puzzle piece fitting in certain region, and there are again millions of chemical compounds that are already out there. This problem is called repurposing an existing drug. Instead of designing a completely new chemical compound, you may also try to see whether existing drugs can help cure some other diseases by trying to find relationships, these docking between existing chemicals and protein structures. I'm going to skip these experimental techniques for structure determination. I mentioned some of them already. You may now have X-ray crystallography, nucleic magnetic resonance. These are experimental methods to determine the protein structure. And many of them are indirect methods. We cannot visually see the protein structure and photograph it like this. We just shoot some rays to it, like X-rays or some photons, and we look at diffraction patterns, and we look at the images we get, and we collect them. Those 2D images are collected to each other together, like those X-ray diffraction patterns, to give us clues about what kind of a three-dimensional shape may have produced those patterns. And by using really algorithms and programs, we then infer all these structure, even the experimental techniques, we actually infer the protein structure with really high accuracy at the end. So X-ray, this is X-ray crystallography. I'm skipping the details of these things. And let's come to our first computational problem that I will, as I said, talk about three main methods for the protein folding problem. And I will also share with you the article about this deep fold, which was a really big news in recent months, which deep fold method structure prediction. Yeah, it's not default actually, AlphaFold. Yeah, sorry. I named it incorrectly. So AlphaFold, a solution to a 50-year-old grand challenge in biology. Right now, in this slide, what we're seeing is this Levinthal's paradox and this problem that I have on these slides for many years. It was really, I was talking about as the protein folding problem as one of the unsolved problems. Now, even the people who made the CASP competition state that we may consider this problem solved now. Okay, so that's bad news for people who try to still solve this problem, but it's good news for science, good news for humanity, a solution to a 50-year-old grand challenge. It was one of the 11 or 10 grand challenges of the millennial challenges for the Cray Institute. But yeah, now we can consider that as solved. So this competition, CASP, Critical Assessment of Protein Structure Prediction, is a very prestigious competition, which every 2 years different algorithms compete to find the structures of proteins. And AlphaFold has been recognized as a solution to this grand challenge, even by the organizers. So this is really remarkable. So this recognition is really remarkable because it was able to find the shape very close to the ground truth shape, with almost no difference with the ground truth. So it talks about the protein folding problem. So this is a really nice article, and the results of the making of there's a video about this too. And here, AlphaFold 2, look at this. You may consider how this in 2020, how in CASP 14, this AlphaFold 2, why was it considered as a solution? Look how it's been since 2006. We get the score was around, the largest it got was 40%, okay, whatever this GDT score is. Okay, so these are the improvements in the median accuracy of predictions. So these are the median accuracy of predictions. From 2006 to 2016, maybe even earlier than it was just at this range. And in 2018, it wasn't really it was still a breakthrough in 2018, but the organizers did not consider it as a solved problem yet, but they were really interested in this AlphaFold's ability to be able to increase this bar from 40s to 60s. But in just 2 years after that, in AlphaFold 2, it was the bar was raised more than twofold, and the organizers now they accepted that, okay, AlphaFold 2 solved this problem. Okay, maybe I don't know whether they're going to have CASP 15 in 2020 or not. But these are probably the experimental results. So this is a really nice figure. Look at the green one is the experimental structure, And the computational prediction is almost identical in these two cases. As you can see, the computational prediction, they were able to find the experimentally determined structure very accurately. So yeah, this article, the DeepMind, this blog from deepmind.com, I'm going to share this one too. So the problem is, given this, you just provided this sequence, your goal is to predict this shape. And but I will still talk about why this is a difficult problem because of these angular freedom of these bonds having some freedom. The search space was really huge. If you have, for example, 100 residue protein, 100 amino acids, if each if the bond between each adjacent amino acids can take only one of the three positions. Imagine, for example, 45 degrees, 90 degrees, 125 degrees. If you talk about the angle between those, if it's just considered not a continuous angle, just consider a discrete one and only have one of the three possible choices. By changing those conformations, by selecting one of those three options between every of those amino acids, every adjacent pair of amino acids, you would have this many possible conformations. Not all of these conformations will be possible because some of them will lead to clashes, collisions with amino acids with each other. Not all of them are possible, but still it's a lot of conformations. So it's really the search space for possible shapes in 3 dimensions is a really almost infinite search space. Okay, so if you just look at all possible conformations, it will take a lot of years to solve this problem. So it's the AlphaFold doesn't look at all the possible conformations basically. But and also in the cell in nature, uh, also does not go through all possible conformations. The protein sequence, amino acid sequence, folds into its native shape very quickly in nanoseconds. And so it's really a fast process naturally that occurs in the cell. So there are different forces, physical forces that drive protein folding. The hydrophobic collapse, amino acids that don't want to be near water, that don't want to be in the surface, they collapse together to form the hydrophobic core. Uh, and the ones that want to be near water, they form the surface. So this is one force that is believed to make the protein shape. Also, cavities are mostly minimized inside the protein structure. There may still be cavities, the minimum volume disulfide bonds. So, meaning that, for example, if you have cysteine amino acids, cysteine contains sulfur in it, and two different sulfurs, they form a disulfide bond, which is really strong and forms stability of the protein structure. But these are all really, as you can see this, the protein structure prediction problem involves lots of biochemistry, right? The biophysics, all those chemical bonds, the electrostatic potentials. It's really not like a computer science problem mostly, but there are certain techniques which use not only these chemistry information, physics information, but using also similarities of protein structures and sequences. We are going to see that. And also, one difficulty that makes this protein structure prediction a difficult problem is that even a single mutation may make changes in the entire structure. And one of the first molecular disease, sickle cell anemia, is the first identified molecular disease, meaning that the single mutation, the glutamine replaced by valine. So this is this charge which wants to be at the surface. If you replace it with valine, so there happens to be a surface, a protein's an amino acid at the surface which doesn't want to be near at the surface. So it forms, so this one, the sixth amino acid in the beta chain, if it's by a single mutation, if it's changed to valine, the hemoglobin, the hemoglobin cells start to agglutinate they start to stick with each other and form large clusters, and this will result in the blood cells lose their regular shapes. Okay, so this is a blood cell, and they will they will not be able to form their regular circular shape. So this is what sickle cell anemia disease. So mutations, they may change the protein structure. They're really important. And structure prediction, I will talk about three main techniques. Ab initio techniques, the main thing you need to know here is that they don't use protein structures that are known previously. They try to predict the protein structure by just using this physics, chemistry information, and the relationship between hydrophobic amino acids. Uh, and Ab initio means from scratch. This is again Latin from initial, from scratch, from not using any previous information. This is what it refers to. And uh, since the search space is huge, usually ab initio techniques are able to find structure for very small protein chains, like 50 to 100 amino acids. They cannot find a solve for larger protein structures. The most popular protein structure prediction methods are homology modeling and threading. Homology modeling, the idea is very intuitive. If, for example, if you have a new protein sequence, but it shows sequence similarity to a protein sequence that is already in the PDB, protein data bank, which means that if you first do a sequence comparison, and you can do that in BLAST, right? If you go to BLAST, yeah, it's right here, BLAST. If you go here, as the target database, let's do nucleotide BLAST, or if you have a protein BLAST. So imagine we have a protein. Okay, we enter its ID here, I mean, it's FASTA sequence here, and then as the database to search against, I can select here the protein data bank proteins. Why do I select protein data bank protein to search similarity of a sequence to a sequence of a protein in the protein data bank? Because the proteins in the protein data bank, they have also known structures, right? This is still a sequence. BLAST does not do structural alignment, okay? Don't understand this. But when we use the protein data bank, do you think we are using those atom coordinates to do the alignment? No. BLAST is a sequence alignment algorithm. BLAST uses the sequences, the amino acid sequences of the proteins that are in the protein data bank. So it still does a sequence similarity search. Okay, this is very critical. So in homology modeling, we first do a sequence comparison. And if at the end of this database search, if I do a BLAST against protein data bank proteins for my protein sequence, and if I see that at the top, I get a hit for a protein data bank protein, for example, and I have 60%, 50% similarity, sequence similarity to that one. This is a really good starting point. That protein data bank protein that I have found, I can then just grab its structure and then use it as a template to build to predict my structure for my new sequence. So there will be differences. It will not have exactly, so just 50% sequence similarity does not mean that they will have the same structure, exact same structure. There will be differences, but parts that align with each other, usually if this is like the rule of thumb, if you have more than 50 or 60% similarity, homology modeling is going to give good results. But there is this twilight zone between 30% similarity or 30 to 40% similarity, sequence similarity, which is called the twilight zone in terms of sequence similarities. That will be difficult to use homology modeling. But you can still use certain parts. If certain parts have really good matches, you can use them in your prediction as your template. So that's the idea of homology modeling. There are homology modeling servers. If you homology modeling server. Civis model. Yeah, Civis model is one of the most Uh, yeah, here as you can see, this the SARS coronavirus proteins, they also appear here. So civis model is a fully automated protein structure homology modeling server. So it means that you just, when you start modeling, just like blast, you'll just paste your sequence here, and then uh, you first search for templates. Search for templates is that sequence comparison part. So it probably does it may be using blast in the background and do the same protein data bank search, database search to search for similar sequences. And then when you click on build model, it's going to give you the predicted model completely automated. Okay, so that's homology modeling. Now, what if we have a long protein? Imagine you have a protein with 1,000 amino acids. So ab initio techniques is out of the picture. And that 1,000 amino acid long protein sequence, you do a blast search against the protein data bank, and the top hit you get contains only 5% sequence identity. So which means that the second option is also out of question. With only 5% sequence similarity, you cannot do homology modeling. It's not possible because you have to, in order to get a template, you have to first find a protein structure with a protein sequence with known structure and have considerable similarity to that. If your new protein with 1,000 sequence and doesn't have any sequence identity, since it's a very large protein structure, you cannot use ab initio. Since there is no sequence similarity, you cannot do homology modeling. The only option that remains to you, but still you shouldn't feel so desperate in that case, because you can still use the threading method, the protein threading method, which I call the Cinderella method. If I have some cables here, I want to show you what I mean. Let me see if I have some thread. Oh, actually, I have I have a, yeah, I use this small puzzle when I'm using protein. I'm talking about protein structure in face-to-face usually. So this may be like the native state of our protein, like a minimum volume, like a cube. Certain surface amino acids are at the surface. So imagine this is its native shape. So the amino acid sequence is this. Okay, so this is this amino acid sequence, as you can see, unfolded. So how do you fold it and make it so in its native state is going to be that cube, and it becomes cube in the cell very quickly. But here's the protein threading method. So we have a sequence. It has no structural similarity, sequence similarity to any of the known sequences. But imagine we have a library of known protein shapes. This is if somebody tells you, for example, a protein, I mean, this chain is going to be either this shape, it can be either this shape, okay, so this one, so they have different shapes, or it may be this shape, okay, which is like linear pen shape. So, or maybe another one. So if you have your protein structure, if you have your protein structure universe represented by these known shapes in the protein structure universe, so this linear chain of amino acids is going to be one of those four things. So the protein threading proceeds like this. It tries to threading basically says that it tries to thread this sequence on top of an existing shape. Okay, it tries to align a sequence uh, against the structure. So it tries to, that's why I call this like the Cinderella method, because it tries to fit this shoe onto Cinderella's foot. So, and this is a difficult problem by itself, because the sequence of amino acids, you want to fold it around a known shape. You want to make it similar to a known protein shape, there are millions of ways to do that. Maybe this problem itself, I mean, do we really make the problem simpler? Threading problem by itself is very difficult, but uh, it's not as difficult as trying to predict its shape from scratch. You just try to see whether if this uh, unfolded string of amino acids, if it was folded, that's going to have this shape. If it's folded like this, for example, do these amino acids prefer to be in this shape? Or do these amino acids, when you try to fold them against the stapler, do they want to be in this shape? So you just try your protein sequence against the known shapes in the protein structure universe. Therefore, protein threading actually requires a fold library. And if this fold library contains all the known shapes we have in the protein structure universe right now, and if this covers the protein structure universe exhaustively in a nice way, it means that you're looking at all possible options. You have all the girls in town who may be Cinderella. If you miss the actual Cinderella, then probably you will be have an incorrect prediction. But if you have all the structural universes exhaustive that you have, this threading may give really good results. So that's the idea of threading. Try to align a sequence, try to see how it's going to prefer a shape like this, or a shape like that, or a shape like this pen. Okay, so it's that's the idea of protein threading, and it gives you a way to predict a structure even without the absence of sequence similarity. So I actually mentioned the main details of all these methods, but in the last couple of minutes, I will just give you some small details. But this is the detail that we are going to be discussing. Actually, we're not going to go into the methodological details of all these things. So for ab initio models, there are some simplifications, assumptions in order to build those. There are some lattice models, for example. They try to solve the protein structures in 2D, for example, by trying to first find how that amino acids are going to be located with respect to each other, whether they're going to be close. If you know, for example, every amino acid you can label as hydrophobic or polar. Okay, so this HP model, hydrophobic polar. For example, arrange your protein sequence in a grid like this so that hydrophobic interactions between non-neighboring, for example, this is not a hydrophobic interaction because they're already adjacent on the sequence. There will always be adjacent. You're not going to break this bond between them. They're adjacent amino acids, so this bond is always between them. But look at this, the dotted line between this amino acid and this amino acid. This is dotted because they're not neighbors in the sequence. They came closer in this 2D space, so this is in hydrophobic contact. So this is a nice puzzle problem. Can you arrange the sequence in such a way that the number of hydrophobic contacts are going to be maximized? So here we have 1, 2, 3, 4, 5 hydrophobic contacts. Can you, for example, it may be a good puzzle for you. Can you try this sequence in unfolded manner? It will just basically saying that the first one, fourth one, sixth one, seventh one, ninth one, 11th, 12th, 14th, 15th, and 18th one, and this 19 amino acid one is hydrophobic, folded in such a way that they will not be, no two amino acids are going to occupy the same space. They cannot collide with each other, and just arrange it in such a fold that the number of hydrophobic contacts are maximized. So this is how ab initio models, some of them work. And this is also, this simple looking puzzle may be a difficult problem. It may be NP complete, which means that nobody has known an efficient solution that works in polynomial time for that. There are different optimization methods, search methods. We search the universe by some search space by greedy techniques, and this is Also, we made certain assumptions, like if you put it in a grid with all the only angles you're going to allow in between them is either 180 degrees or 90 degrees. You may have also some models with 45-degree lattice, off lattice models, three-dimensional models. So this is, and you may also use, instead of hydrogen bonds only, you may use some energy functions. So it's really difficult to, since the search space is huge, ab initio methods only work for small proteins. And this is a difficult problem because there are global interactions between amino acids that are far away. And that's all you need to know about ab initio models. I will stop here today. Okay, on Thursday, we are going to finish homology modeling and protein threading, and we will talk about structural alignment. And I hope to finish it by then. Probably I will finish talking about protein structures on Thursday. I will quickly go over the remaining set of slides. But yeah, that's it for today from me. If you have any questions, I will be happy to answer them. I will stop recording now.